Hi all, my name is Professor Charles Walker of the Molecular, Cellular, and Biomedical Sciences Department at the University of New Hampshire. Today, my colleagues here at UNH and I will discuss with you the largest protein structure in the eukaryotic cell, the nuclear pore. Following synthesis in the cytoplasm, all proteins that function within the nucleus must be imported. This enigmatic process occurs at a rate of 1,000 proteins every second. In the cytoplasm, there are receptors that recognize proteins destined for nuclear import. These receptors chaperone proteins first to and then into the nucleus. Import takes place through numerous pores in the nuclear membrane. In this video, we begin within the cytoplasm of a cell and focus on one of the many pores embedded in its nuclear membrane. In this side view of a single pore, we see that many structural proteins in purple make up this massive structure. Each pore contains a group of spaghetti-like protein filaments in blue that must be negotiated during import. The blue massive filaments in the pore seems to present a formidable challenge to entering the nucleus, but it is also the mechanism that selectively permits proteins to enter. Receptor proteins and proteins destined for nuclear import bind to filaments extending from the pore into the cytoplasm. These protein complexes can then enter the massive filaments in this pore. How this complex of proteins negotiates these filaments remains a mystery, thus the impression of chaos in the film. Once within the nucleus, the receptors and nuclear proteins separate after exiting the nuclear basket. We will propose some strategies for accomplishing import of nuclear proteins based on current knowledge of the biology, chemistry, physics, and mathematics of nuclear pores. See if you can think of more.
Hello, my name is Karen Graham, and I'm a professor in the Department of Mathematics and Statistics. Before we examine the process further, it is often helpful to get a sense of the infrastructure of the nucleus and the scope of the process. The shape of the nucleus can be thought of as a sphere. To get a sense of the scope of the import process, we could ask, what is the number of proteins per second that are imported by the nucleus? To answer this question, we would need to know the number of pores on the nucleus and the number of proteins that are imported per second by each of these pores. We know that 1,000 proteins are imported per second per pore, so we need to determine the number of pores on the nucleus and then multiply by this rate. In other words, the number of proteins imported by the nucleus per second is equal to the number of pores per nucleus times 1,000 proteins per second per pore. Unfortunately, we do not know the exact number of pores for a given nucleus, but using averages, we can get a handle on the maximum number of pores possible for an average nucleus. To do this, remember we are thinking of the nucleus as a sphere. We can determine the surface area of our idealized sphere using the formula surface area is equal to 4 pi r squared, where r is the radius of the sphere. Let's assume that the diameter of an average nucleus is 10 micrometers. Then the surface area of the average nucleus is 4 pi times 5 squared, which is equal to 100 pi, or approximately 314 square micrometers. Now, to determine the maximum number of pores that could be located on the surface of our idealized sphere or nucleus, we would need to know the area of the pore complex. To calculate the area of the complex, we can think of the complex as a circle and use the formula area is equal to pi r squared. The average pore complex has a diameter of 100 nanometers and therefore a radius of 50 nanometers. Using the radius of 50 nanometers, we have the area is equal to pi times 50 squared, which is equal to 2,500 pi, or approximately 7,850 square nanometers. So the average nucleus has a surface area of approximately 314 square micrometers and the area of the average pore complex is 7,850 square nanometers. To determine the maximum number of pores on our average nucleus, we divide the surface area of the average nucleus by the area of the average complex, which is equal to 314 square micrometers divided by 7,850 square nanometers. But the units of our calculations are different, so we first need to convert the same to the same unit. Now we know that 1 square micrometer is equal to 10 to the 6 square nanometers. So 7,850 square nanometers is equal to 0.00785 square micrometers. Plugging all of this information into our equation, we have that 314 square micrometers divided by 7,850 square nanometers is equal to 314 square micrometers divided by 0.00785 square micrometers, which is equal to 40,000, which is the maximum number of pores. Wow, that's a lot of pores. But this is a maximum number, and we know that the average nucleus has far less of its surface covered with pores, probably only approximately 30 to 40%. So let's say it has 40%. This would mean the number of pores is more like 40% of 40,000, which is equal to 16,000. Still a relatively large number. Using this number of pores and the rate of 1,000 proteins per second per pore, we have 16,000 pores per nucleus times 1,000 proteins per second per pore, or 16 million proteins per second imported by the average nucleus. It's unlikely that this number of pores would ever be reached for a given cell. It all depends on the number of pores on a particular nucleus, but this gives you a sense of how one could calculate the number of proteins per second that are imported per nucleus. Bottom line, there is a lot going on.
Hi, I'm Professor Bauer from the Chemistry Department. Let's take a look at one of those many nuclear pores and consider what we know about how materials are allowed to move through the pore and across the nuclear membrane. Remember that the cell has an outer cell membrane as well. The outer cell membrane also has pores in it, but these pores are about 10 times smaller in diameter than the nuclear pores, because most of them are conduits for small molecules such as water or simple ions. In contrast, the pores in the nuclear membrane are huge. It's like the difference in width between a straw and the mouth of the plastic cup that the straw sits in. Open gaping holes this large would allow unnecessary molecular material to diffuse into and out of the nucleus in an unregulated way. What has been found is that the nuclear pore is partly occupied by protein filaments that act like a pile of cooked spaghetti. The spaghetti strands have long segments containing amino acids with nonpolar hydrophobic side chains, in particular phenylalanine and glycine, shown here. These filaments seem to fulfill the same role as the hydrophobic interior of the membrane. They act as a barrier to movement of water-soluble materials. But like cooked spaghetti, they are slippery because the only type of interaction between them is the relatively weak nonpolar interactions. Given this structure, one can imagine fairly large molecules slithering into and through this tangle. Getting into the pore seems to require a chaperone protein to attach. Getting out seems to require removal of the chaperone with energy input from hydrolysis of GTP, a relative of ATP. Things get fuzzy in between. Exactly how the chaperone assists its baggage to get into and move through the tangle is not well understood. Perhaps the chaperone has an enhanced attraction to the spaghetti and slides along, being pulled gently from one strand to the next. Perhaps the spaghetti is like a nonpolar zone, and the chaperone protein just dissolves better in it and eventually moves randomly within that zone till it accidentally comes to the other side. Perhaps the chaperone causes the spaghetti strands to part and allow the baggage a window to move through. We can test these hypotheses by exploring questions like, if this mechanism is the way it works, we should expect to find the following types of molecular structures on the chaperone protein. Or, we should expect the time it takes to get across the pore to be X milliseconds. Or, we should expect to find N molecules of GTP hydrolyzing to provide the energy needed. To narrow things down, it is necessary to design experiments that allow measurement, visualization, or manipulation of structures, dimensions, or durations.
Hi, I'm Professor Dawn Meredith from the Physics Department. I'd like to pick up on Professor Bauer's discussion by focusing on two of the possible passive mechanisms which he mentioned. One mechanism might be that the chaperoned protein just dissolves better in the spaghetti than the unchaperoned protein. The other is that the chaperone causes the spaghetti strands to move apart and make room for the protein. Both of these sound like chaperone-enhanced diffusion. That is, chaperones increase the protein diffusion constant by making motion through the spaghetti easier. How might we test these ideas? First, we can check if energy is required, other than for attaching or releasing the chaperones. If so, this is not diffusion. Diffusion does not require energy input from the cell, which is why it is a useful transport mechanism. Second, diffusion always requires a concentration gradient, so we could check if there is a concentration gradient for the protein, with a concentration lower inside the nucleus. Third, if it is diffusion, we would expect motion through the pores in both directions for a single protein. That is, at each step, a protein is equally likely to move toward or away from the nucleus. We can put a tracer on the protein, and if we see the motion in only one direction for a single protein, that would mean that it is not diffusion. Fourth, are there other molecules that we would expect to diffuse more easily than our proteins, and are they moving through the nuclear pore? These easy-to-diffuse molecules would be much smaller than our proteins and would have a larger concentration gradient. The proteins that are being transported through are quite large, 50,000 Daltons, and there are many much smaller molecules, such as glucose at 180 Daltons, that we would expect to be able to diffuse quite easily in spite of the spaghetti-clogged pore.
So to summarize and think ahead, let's consider how important this process is to each of your 70 trillion cells. Since proteins that function in the nucleus must be made in the cytoplasm and then imported, an efficient and highly discriminatory process is essential. We know that cytoplasmic receptor proteins seek out newly synthesized cytoplasmic proteins that belong in the nucleus, and that there are pores more than sufficient in number to get them there. What we still find enigmatic is the exact mechanism by which nuclear-bound proteins negotiate the pore to make sure that only the right proteins enter. We present several options for this based on our current understanding of the chemistry and physics of the spaghetti of filaments blocking the pore, and more than one of these mechanisms may be involved. Perhaps you folks will accept the challenge and combine your knowledge of biology, chemistry, physics, and mathematics to uncover the mechanism.